Great, thank you very much. Grazie a voi per posso parlare italiano oggi. Otherwise, we'd be in trouble. So, thank you very much. I'm a human interventional radiologist, board certified in the United States with added qualification for interventional radiology. And I'll give a little bit of history of my background. Currently, I'm the Chief Medical Officer for Epic International, which is a holding company for Imaginalis. So we're all one big family, but just so you know, I am on the payroll. A number of people I would love to thank today. Most importantly is my friend, mentor, and colleague at UC Davis, Dr. Bill Culp. He's a veterinarian, soft tissue surgeon, orthopedic, oncologic surgeon. I'm in great company today also with our key opinion leaders. We've had some excellent lectures today, and we have more to come. So it's always great to be in good company. Thanks to Vieri and his team for actually putting on this wonderful Congress for us. And also my inspiration, although they are both very humble, are wonderful international veterinary application specialist team. Can't do it without them. They are actually showing me the way for our human products, which are coming out shortly. And of course, a lot can be said about employee number one of Imaginalis, Leonardo Manetti, but maybe not so much should be said, so we'll leave it at that. But Leo, it's been an amazing couple of years with you, so I thank you very much.
Thank you.
And last, well, of course, everyone at IMG, the engineers, the production line, everybody there is fantastic. So thank you to them for making it. It's not possible without them to make the machines. And I need to give a shout out to my American colleague who's sitting here in the second row, because he was the first one I came in contact with Epica, so I completely blame him for being now part of this crazy Italian company. 17 years ago, when I came back to my hometown in Sacramento, California, I was approached by Dr. Culp, who is fresh from University of Pennsylvania. And he came to me and he said, I want to do interventional radiology, minimally invasive procedures. Can you help me? And of course I said yes, and over the last 17 years I have brought him every expired piece of equipment I could find. Once a month we would be doing some sort of procedure in the interventional suite, and where we started was actually in the basement of the horse barn, where cardiology did a lot of their work with this really clunky C-arm that you can see behind Bill. With really awful equipment. And of course, the very first case we do is a limb AVM with Onyx. So we're using equipment that costs $1,000 for a 3 cc vial in the basement that's not sterile with made up tools. And so we kind of developed it along the way, but this is where we started. He is mostly a small animal vet, so that's at UC Davis, it's a huge service, and he called me for absolutely anything and everything that he would get consulted on, whether it was vascular, biliary intervention, urinary intervention, and every oncologic procedure that he wanted to do. We used every imaging modality at his fingertips, which includes fluoroscopy primarily, CT, ultrasound, and we do a combination in his operating room open. So he's primarily vascular, so he treats a lot of liver shunts, he treats a lot of AVMs, and soft tissue tumors. So as we developed more and more of these procedures and grew our experience, we then expanded to larger animals. And I distinctly remember this first case. It was a horse with a very large staghorn calculus you can see on the ultrasound to the right. And they prepped the horse. They brought me in and put a ladder next to the side of the horse, and they handed me a 16 gauge needle. It was about this long. Handed it to me. I got up on the ladder and everybody stepped back. I take the 16 gauge needle with the ultrasound and I start to advance it into the kidney, and the horse kicks underneath and kicks the ladder out from underneath me. So it was quite an experience, but nonetheless we were successful in getting a wire down, and they did a transperineal approach to do the nephrolithotomy. And then from there into the exotics. So this is us at the zoo. We've worked on lemurs, we've worked on Sumatran tigers, and the great thing about Bill, as you can see in the upper left corner, he's like, I have to tell you something. There's too many people in the operating room here today for us to have a weapon to put the animal down if it wakes up from anesthesia. So you see those two doors over there? If the animal wakes up, you run. I said, you got to be kidding me. He says, no, I'm not. This is the zoo. We usually keep a 45 here so we can put the animal down. But he's a tremendous guy, a great friend, and he loves to push the boundaries. So it's been a great experience. And because of him, they started a veterinary interventional radiology and endoscopic society called VIRES. And he asked me to be speaker the very first year. And at that meeting in 2015 is where I met Ron Tibbett. I walked by the Imaginalis booth, saw the images and the technology, and from there it's history. I became involved with the advisory board, became the clinical director, and now chief medical officer. So this is just a variety of the type of image-guided procedures that Dr. Culp and I do at UC Davis. I used to scrub in on every case. Now he's facile. I just show up and drink a coffee at the back table while he does the procedure with his residents and fellows. But these are all high-level human interventional radiology cases. We're doing a tremendous amount of work, mostly with C-arm, but now he wants to involve CT. We've done a couple of our microwave ablations in the 64-slice CT scanner that they have at UC Davis, and it disrupts the whole workflow. Radiology hates it. The OR staff hates it. They have to take the animal and all the equipment down into the radiology suite. No one knows how long it's going to take. Meanwhile, the regular workflow is getting backed up for all the diagnostic CT. So it just makes sense to have CT imaging now at your hands in the operating room. I have 9 co-authored papers with Bill. He's prolific. He's got 174 peer-reviewed publications. And we hope to have many, many more so that we can join what's going on in Greece. And hopefully we will have some level 1 data for you all next year when we come back and do this meeting again. So here we are. VIRES was in 2015. 10 years later, we now have the Vimago in Bill's dedicated operating room, which is now actually in the OR. It's a very large space, so you can see we have his brand new GE OEC 9900, the Vimago in the background in the transport mode, and then his existing Steely table, which is an interventional table, mushroom tabletop that he can move easily with his hand. Anesthesia is at the foot of the bed, and then the other piece of equipment that we have, which is very important, is a mobile lead shield. So you can stand behind it if you're doing injections during CT or even long fluoroscopy cases. Let's see if this will work. So Bill called me up and said, now that we have the GT30, why don't we do some CT angiography? I was like, great, that's the worst possible first case we could do. But he wanted to get a good feel for the vascular anatomy. Let me see if I can just pause it right here. Okay. So with AVMs, and I apologize, I don't want to insult anybody's intelligence, but the whole goal here is to kill something called the nidus, which is actually a soft tissue component of this tangle of vessels that has an arterial origin, a network or a spider web network of all these small vessels that is hormonally active, and you need to kill that nidus so that it stops shunting blood and does not continue to grow. So as you can see, in a 2-dimensional fluoroscopic view, this is incredibly difficult to tell where you're going. Where do I need to put my catheter to feed this nidus? Why not have some sort of cross-sectional imaging like CT, better yet, 3-dimensional imaging? So he thought, let's use the Vimago, let's get some 3D images, and we can then map out this whole lesion, feel much better about treating it. This was a pit bull that was 35 kilograms. We went in through the right femoral artery. Dr. Culp does a cut down, isolates the common femoral, actually ligates the vessel when he's done, completely cuts it off. It still to this day boggles my mind. Run a catheter up under fluoroscopy into the right common carotid artery to do a first injection, which is what you're seeing here on the left. And he typically uses a 4 French or a 5 French Cobra, depending on the size of the femoral artery, and then works coaxially with a very small 1 millimeter or 3 French microcatheter through that to do the treatment. So you're always maintaining your access within the feeding vessel, and then you can use another tool to do treatments in different areas, because there are a number of blood vessels that we'll need to address. And then for these, depending on the speed or the flow through the lesion, we will treat it with glue and lipiodol or thiodol, which is a poppy seed oil, radio-opaque material. And you can mix the two to make it a thick embolic. So it's a liquid embolic that will go out into all of the small branches rather than particles or coils. And so this was the first time we'd ever used a combination or multimodality approach to a procedure. So we're using his C-arm, and we're also using the GT30. And what that looks like is this. So the C-arm now is moved out of the way so that we can get access to the neck should we need. The GT30 is just pushed back out of the way so we can bring it in, and we can also move the table in and out so that we can do the scan. Now, one thing to note, the Vimago in its normal scanning configuration can do 7 stacks. In the transport mode, where the gantry is turned 90 degrees, we can only do 1 stack. So, but for Bill and for our field of view, 1 stack is plenty. So we have the 4 French catheter into the common carotid. We have the 3 French microcatheter up into the feeding vessel. And we did CT angiography. And this is just another way of what we've done in human medicine since 2003. It's called C-arm CT. And Siemens came to Washington University in St. Louis, where I was training, and said, we need some opinion leaders to come try this. We want to take our flat panel C-arm and turn it into a CT. So this technique has been around for a long time. And the only problem with the C-arm is that you can rotate, at that time, 220 degrees. So you couldn't go all the way around. You're missing a whole gap of data. And so the images, when they come out, are very grainy. They're very poor quality. But it's enough to help you map or to know what your perfusion bed will be. So it was still useful, but it was a lot of work. It took 8 minutes to reconstruct the image. You had to put the patient's arms over their head. You had to connect to the injector and do a very slow dilute injection. So it really changed workflow, and it didn't take off until maybe the last 5 to 8 years now, when people are doing more oncologic work in the liver, where it's very important to know what vessels you're treating with radioembolization or chemoembolization. So for this case, we used 12 cc's of Omnipaque 300. We diluted it to 50%. We put it into a 20 cc syringe. Bill stood behind the lead shielding between himself and the Vimago so that he wouldn't get radiated. And over the 24-second scan around the neck, he injected the whole time this dilute contrast to give us this image. And within 1 minute of the scan, we had this 3-dimensional image that we could then work off and plan and see, oh, we've got 2 feeding vessels here. One coming off the inferior thyroidal, and I think the other one was the pharyngeal, ascending pharyngeal. But without this information, looking at the 2-dimensional fluoroscopy, it was very difficult to know where we had to go, and it would require multiple catheterizations, multiple rotations to figure out where we need to be. And so we estimated that we saved ourselves about an hour of procedure time. And with that, all of the radiation from fluoroscopy that would go with it. And this is him doing the injection with the microcatheter in one of the vessels. And to Jan Kuka's point of how important it is to be able to see where the monitors are while you're working on the patient is critical. So placement of, we're not in the gantry, we're away from the gantry because we're treating in an area that is under the C-arm, not where we're working. So if you're working in the gantry and you have to lean back to look at the monitor, Dr. Kuka's point is very well taken. What you're seeing on this injection is the combination of glue and Lipiodol, and the glue is N-butyl cyanoacrylate, so it's super glue. You get a medical grade, it'll cost you $1,000 for a little tube. You go to Home Depot, it'll cost you $1. But as you can see, even under fluoroscopy, okay, we see that one main feeding branch, but we don't see all of the nidus. So we're still not sure how much of the lesion we've treated. But as we fill it, there comes a point where we have to stop injecting the glue, otherwise it will reflux and go to other branches where we don't want it to go, particularly the brain, since we're working in the carotid. One other thing I should say is what helped was to clamp or occlude the dominant outflow vein. So now we're pinching the outflow to slow the flow through so it wouldn't go through to the lungs. So you close off the dominant outflow vein, the flow into the malformation is slower, and you get a better fill of the glue. And then post-contrast, we repeated the CT scan, no contrast injection. And I'm sorry, I was very excited when I was taking this video, so it'll kind of drift off to the side, but then I swear it will come back. And one of the unique things about this viewer software is the ability to fuse the CT image over or overlay the image onto the actual 3D model. And so you can see it, here it is in 2D, and the tremendous fill we actually have of the nidus, we can now see on 2D and 3D. This is a very powerful tool as someone who does procedures. You can remove the clip plane and isolate the lesions itself, and now you can see we've successfully treated the superior nidus and also the inferior medial nidus. So now we have good confidence that we've treated the areas that we needed to treat. We know that we're done with the procedure. We don't have to go back and do more injections, more x-ray, longer surgery time, longer anesthesia time. We're very confident that we have successfully treated this lesion. So now, where do we go from here? Recently, Imaginalis has expanded their product line to include a particular model called the Centauri, which you can see here on the right side. I'll have more information. There's likely some in your folders. But this has a much better design for intraoperative use, where there's no end and the surgical table can come into the field. Now we can index the gantry and get at least 5 stacks. So it doesn't handcuff us with space. It doesn't handcuff us with only being able to do one set of images, one stack. And it allows us to actually slide it right up next to the surgical table. So in Europe, where there is a very small operating rooms, the Centauri, which can also be used for equine, is also a great option for small animal surgery. And I'm sure with Dr. Kuka's help, we will improve the fluoroscopy for increased utilization for the GT30 family, the Vimago family. And what I would love to see is for Bill to now push his C-arm into the corner and only use the Centauri or the Vimago product to do both fluoroscopy and CT. Bring it in, do fluoroscopy, CT, move it out, do the surgery, bring it back in. It's like a hybrid operating room for humans where you have the CT or an MRI built into the wall of the operating room, and it slides out on a track to do the imaging, and then it slides back out of the way. This is a fantastic approach for intraoperative use. And then we can think to use the 3D modeling for biliary cases. The biliary tree is also very much like the vascular tree in that it's tortuous and difficult to see in 2 dimensions. So having the 3D modeling will be extremely helpful. And then we mentioned also here ablation and biopsy, and really that is where the innovation comes in, which just recently the Navigator has been released to have the 3-dimensional capability to see your lesion and use it for treatment, for real-time road mapping, to reduce the amount of radiation that you use, to be more precise, to reduce your operating room time, to be sure of where your catheters and your needles are, and if you're going to ablate other structures that are nearby that you need to be careful of. And these are all value-based propositions, which makes this technology very, very useful in the operating room. And then eventually multimodality fusion imaging. Bring in MRI, fuse it over the CT. Bring in CT, fuse it over the fluoroscopy. What can we do? So there are things that would be great to look at. And ultimately, if you have more confidence in your outcomes, your patients will do better. Your clients will be happier. And to keep it short, thank you all for allowing me to be here. It's always a pleasure to come to Italy. Take any questions.